How centrosomes help to create multicellular animals via hierarchical endosymbiosis, and the evolution of animal cells from viruses. Step 1. Virus to bacterium. What kind of virus in bacterium? Transposons and archaea bacteria. Wait, I'll explain. What is a transposon? It's not considered to be a virus per se. Viruses have their own capsules. This protects their nucleic acids when they're outside the cell. Transposons have no capsules. They can only exist within the cell. They are called jumping genes. They can move from one area of a chromosome to another and reinsert themselves there. They can also be inactivated so they no longer jump. They exist in archaea bacteria as well as animal cells. Both animal cells and archaea have multiple origins of DNA replication. Both cell types contain chromatin in their chromosomes. Chromatin is DNA wound around and compacted by proteins called histones. The basic thread of chromatin is 30 nanometers in width. Nm stands for nanometer, which is 1/1000th of a meter. DNA itself is only 2 nanometers in width and extremely fragile. A 30 nanometer thick chromatin circle laid flat against itself would be at least 60 nanometers in width. My model suggests that fragile sites between sections of chromosomes may be connected by such circles. Scientists assume archaea bacteria are the original ancestors of animal cells. Time for some diagrams. In this model, a DNA replicon is a double-stranded circular DNA capable of self-replication when its origin of replication is attached to another origin and modified for replication. Enhancers may allow two replicons to bind together at their enhancer sites to create a double origin of replication. Enhancers can enhance promoter regions on DNA which create RNA. In my models, enhancers do this by first creating a double origin of replication, which is then transformed into a promoter region. The process can continue indefinitely, generating huge circles of DNA. Our differentiation can take place by inactivating transposons. For more details on modification and activation of double origins of replication, promoters, and splice sites, watch the following presentation. Step 2. Centrosomes. What are centrosomes? Centrosomes are used in animal cells during mitosis. They assist in pulling apart chromosomes using spindle fibers. One set of chromosomes goes into each new daughter cell. Okay, back to the diagrams. This section is about my radial model of centrosomes. As you can see in figures 6 and 7 and 10, there are layers or radial concentric rings of chromatin that you can see in all three of these photomicrographs, which were taken at 400x magnification using phase contrast microscopy. And so I am basing my radial models, which I will be putting forth here, based upon these photomicrographs. Each ring houses chromatin destined to become 4 chromosomes. Each future chromosome is shown in different colors. This slide shows 4 gaps between future chromosomes in ring number 1. There are similar gaps in rings 2 and 3 as well. Duplication of beads in centrosomes is complete. Their counterparts are behind the plane of the drawing. Chromatin beads are drawn together and separated from other chromosomal beads. Centrosomes are pulling away from each other in front of and behind the plane of the drawing. The chromosomes are formed, and with them come restricted areas called fragile sites. The chromosome spindles are extended. The chromosomes are pulled to the metaphase plate by the centrosomes. All of these photos can be explained with my models, except for the redacted ones shown in the red rectangle. What the heck is going on here? There's no centrosome. Was this derived from a chromatin bead on a ring? If so, what is it? Looks like a miniature model of a ring. Or could it be something else entirely? Instead of a tiny centrosome surrounded by outer rings of chromosomes, perhaps this is an entirely different structure. Perhaps it's a complex replicon cluster. When the microphotographs shown in this presentation were taken, the cells used in the study were supposed to be dormant. They were supposed to be locked into one position of the cell cycle. That doesn't seem to be evident from the photos. Instead, they seem to be moving slowly through the cell cycle, hence the many types of structures presented here. Step 3. Fragile sites. What are fragile sites? Fragile sites are weak spots found on chromatids. Chromatids are the arms of chromosomes. In my models, chromatids are composed of subchromatids. These subchromatids are connected together by fragile sites. Learn more by visiting PowerPoint XV. Near the bottom of the post, click on XV. Time for some diagrams from PowerPoint XV. This slide requires some background information. What does it mean when it says larger chromosomes from smaller chromosomes? Again, a fuller explanation can be had by watching PowerPoint XV, but I'll keep it short here. What that statement means is that chromosome structure has evolved from combinations of smaller chromosomes over time through a process I call hierarchical endosymbiosis. A mouthful for sure. In this slide, one larger chromosome has evolved by fusions of 3 smaller chromosomes, generating 2 fragile sites between them. Subchromatin separation at a fragile site. Let's focus on that one fragile site. By stripping away everything but the DNA, and then all the DNA but the fragile site. Enlarge it down to the DNA level. Fragile sites are where a minimum of 2 origins of replication join together. Origins can separate after damage by radiation, mutagens, or during chromosome diminution, or even by physical damage during the stress of mitotic separation of chromosomes. Leads to all kinds of genetic diseases. Not all double origins become fragile sites. Otherwise, chromosomes would just fall apart. They get their strength from at least 3 sources. Parallel double origins, strength in numbers, amount of protein available for packing, level of overall compaction. Enough already. Why should you care? Simple. The more we understand about ourselves, the better off we are. Every disease that affects us is because something is wrong with our cells. And you can help in the usual ways. Share, comment, suggestions, fund a small project. How? Contact me.